Hello and welcome to the Hospital Near Me Medical Career Training Channel. Welcome to our educational series where we unravel the mysteries of biology. Today we're diving deep into a range of fascinating topics, from the basics of DNA replication to the critical functions of the human liver and respiratory system. Today we are working on the Medical College Admissions Test, or MCAT, to get you into medical school. Today's featured hospital is Albany Medical Center Hospital, located at 47 New Scotland Ave, Albany, NY 12208. Phone number is 518-262-5521. Admissions phone number is 518-262-5521. Schaffer Library of Health Sciences phone number is 518-262-5530. Columbia Memorial Health 71 Prospect Ave, Hudson, NY 12534 phone number is 518-828-7601. Patient billing 518-828-8051. Their general phone number is 518-262-3125. Do you need to talk to someone? Get confidential help through talking with a full services professional listening company. Reduce your stress and increase your peace. Get problem-solving help today. Click on the link in the description that says, "Let me help you in life." Increase your peace. Get problem-solving help today. Question 1. DNA replication and mitosis. Discuss the process of DNA replication, highlighting the role of the lagging strand and its importance in accurate genetic replication. DNA replication involves the synthesis of two complementary strands, the leading strand and the lagging strand. The lagging strand is synthesized discontinuously in small fragments called Okazaki fragments. The process of lagging strand synthesis is a complex yet highly coordinated series of events that ensure accurate and efficient DNA replication. Initiation. DNA replication begins at the origin of replication, where the DNA double helix unwinds to expose the template strands. DNA helicase unwinds the DNA in both directions, creating two replication forks. Primer synthesis. Primase synthesizes short RNA primers complementary to the DNA template. These primers provide a starting point for DNA synthesis. Polymerase recruitment. DNA polymerase III is recruited to the prime template and begins synthesizing the leading strand continuously in the 5' to 3' direction. Lagging strand synthesis. The lagging strand is synthesized in the 3 to 5' direction, opposite to the direction of DNA unwinding. RNA primers are synthesized by primase along the lagging strand template at intervals. Okazaki fragment synthesis. DNA polymerase III adds nucleotides to the 3' end of the RNA primer, synthesizing short fragments called Okazaki fragments. Each Okazaki fragment is typically around 100 to 200 nucleotides long. RNase H and DNA polymerase I activity. RNase H removes the RNA primers from the Okazaki fragments. DNA polymerase I replaces the RNA primers with DNA nucleotides in the 5' to 3' direction. Ligase action. DNA ligase seals the nicks between adjacent Okazaki fragments, forming a continuous lagging strand. Overall process. The process continues bidirectionally until the entire DNA molecule is replicated. The result is two daughter DNA molecules, each consisting of one original, parental strand and one newly synthesized daughter strand. Understanding the synthesis of the lagging strand is crucial for comprehending the intricacies of DNA replication. The coordination of multiple enzymes and the semi-discontinuous nature of lagging strand synthesis ensure the faithful transmission of genetic information during cell division. This knowledge is highly relevant for individuals studying biology or preparing for examinations such as the MCAT, where a deep understanding of molecular biology is essential. Question 2. Phagocytosis. Explore the mechanism of phagocytosis, a vital process by which cells engulf and destroy pathogens or debris. White blood cells, or leukocytes, play a crucial role in the immune system, defending the body against infections and foreign invaders. There are several types of white blood cells, each with specific functions. Here's a brief overview that may be relevant for the MCAT test. Neutrophils. Function. Neutrophils are the most abundant type of white blood cells and are highly effective against bacterial infections. They engulf and digest bacteria through a process called phagocytosis. Lymphocytes. Function. Lymphocytes are divided into two main types, T cells and B cells. T cells. These cells play a central role in cell-mediated immunity. They can directly attack and destroy infected cells or coordinate the immune response. B cells. B cells produce antibodies, proteins that target and neutralize pathogens such as bacteria and viruses. Monocytes. Function. Monocytes are large white blood cells that transform into macrophages when they enter tissues. Macrophages are essential for phagocytosis, breaking down pathogens and presenting their antigens to activate other immune cells. Eosinophils. Function. Eosinophils are primarily involved in combating parasitic infections and allergic reactions. They release toxins to destroy parasites and modulate the immune response during allergies. Basophils. Function. Basophils release histamine, a compound involved in inflammatory responses. They play a role in the early stages of inflammation and defense against parasites. Understanding the functions of these white blood cells is crucial for comprehending how the immune system responds to different types of threats. The interactions and coordination among these cells ensure a well-regulated and effective immune response, a key concept in immunology that may be assessed in the MCAT exam. The immune system is a complex network of cells, tissues, and organs that work together to defend the body against a wide array of threats, including pathogens, toxins, and abnormal cells. The response of the immune system is highly coordinated and involves both innate and adaptive immunity. Here's a detailed overview at a college level. Innate immunity, barriers and recognition. The first line of defense includes physical and chemical barriers, such as the skin and mucous membranes. Innate immune cells, like macrophages and neutrophils, recognize common features of pathogens through pattern recognition receptors, PRRs. Phagocytosis. Phagocytic cells engulf and digest pathogens, clearing them from the body. This process is rapid and nonspecific, providing immediate protection. Inflammatory response. Triggering inflammation. When tissues are damaged or infected, cells release signals that trigger an inflammatory response. This involves increased blood flow, recruitment of immune cells, and the release of signaling molecules like cytokines. Phagocyte activation. Inflammation enhances the activity of phagocytes, helping to eliminate pathogens. It also promotes tissue repair. Adaptive immunity, antigen recognition. Adaptive immunity is specific and involves the recognition of antigens, unique molecules associated with pathogens. B cells and T cells are central to this process. B cells and antibodies. B cells produce antibodies that bind to specific antigens. Antibodies neutralize pathogens and tag them for destruction by other immune cells. T cells and cell-mediated immunity. T cells directly attack infected or abnormal cells. Cytotoxic T cells destroy infected cells, while helper T cells coordinate immune responses. Memory and long-term protection. Immunological memory. After an infection, memory B and T cells are generated. These cells remember the specific pathogens, providing a faster and more robust response upon re-exposure. Vaccination. Vaccination stimulates the immune system to generate memory cells without causing the disease, offering protection against future infections. Immune regulation. Tolerance. The immune system must distinguish between self and non-self to avoid attacking the body's own cells. Regulatory T cells help maintain immune tolerance and prevent autoimmunity. The immune system's ability to respond to diverse threats is a testament to its complexity and adaptability. The coordination between innate and adaptive immunity ensures a multifaceted defense strategy tailored to specific challenges, a concept essential for understanding immunology at the college level.
The hepatic portal vein plays a crucial role in supporting liver function, and understanding its significance is important for individuals preparing for the MCAT exam. The hepatic portal vein is a major blood vessel that carries blood from the digestive organs, such as the stomach and intestines, to the liver. This venous system is distinct from the systemic circulation because it directs blood to the liver before entering the general circulation. One key function of the hepatic portal vein is to transport nutrient-rich blood from the digestive system to the liver for processing and storage. After a meal, the intestines absorb nutrients from digested food, including glucose, amino acids, and vitamins. These nutrients are then transported via the hepatic portal vein directly to the liver. The liver plays a central role in regulating nutrient metabolism and maintaining homeostasis. As blood from the hepatic portal vein enters the liver, hepatocytes actively participate in processes such as glycogenesis, where excess glucose is stored as glycogen, and gluconeogenesis, where new glucose is synthesized when needed. The liver also processes amino acids, converting them into proteins or releasing them into the bloodstream as needed. Furthermore, the hepatic portal vein serves as a crucial detoxification pathway. It filters out harmful substances, toxins, and potentially harmful microorganisms from the blood before it enters the systemic circulation. The liver detoxifies and metabolizes these substances, ensuring that only purified blood is released into the general circulation. In summary, the hepatic portal vein plays a vital role in supporting liver function by transporting nutrient-rich blood from the digestive system to the liver. This process allows the liver to regulate nutrient metabolism, store excess nutrients, and detoxify the blood before it enters the systemic circulation. A comprehensive understanding of this physiological process is essential for success on the MCAT exam, particularly in sections related to biology and human anatomy. The hepatic portal vein plays a crucial role in supporting liver function by transporting blood from the digestive organs to the liver. This anatomical feature is vital for maintaining homeostasis and ensuring the efficient processing of nutrients and other substances absorbed from the gastrointestinal tract. Blood in the hepatic portal vein is rich in various products of digestion, including amino acids, glucose, and lipids, as well as absorbed toxins and other metabolic byproducts. This nutrient-laden blood is delivered directly to the liver for processing before entering the systemic circulation. Key points about blood in the hepatic portal vein. Nutrient processing. The hepatic portal vein delivers blood containing nutrients from the intestines and stomach to the liver. This includes amino acids from protein digestion, glucose from carbohydrate digestion, and lipids from fat digestion. Detoxification. The liver plays a crucial role in detoxifying the blood. Toxins and harmful substances absorbed during digestion are processed and neutralized in the liver before the blood continues its circulation throughout the body. Metabolism and storage. The liver regulates glucose levels in the blood by either storing excess glucose as glycogen or releasing glucose into the bloodstream as needed. Additionally, the liver processes and stores vitamins and minerals. Synthesis of proteins. The liver is involved in the synthesis of various proteins, including clotting factors and plasma proteins. This is essential for maintaining proper blood coagulation and overall plasma composition. Maintaining homeostasis. The hepatic portal vein is a key component in the regulation of homeostasis. The liver adjusts nutrient levels in the blood, ensuring a balanced and consistent internal environment for the body. Understanding the role of the hepatic portal vein in transporting nutrient-rich blood to the liver is crucial for comprehending the overall function of the digestive and hepatic systems. This knowledge is pertinent not only in the context of physiology, but also in clinical applications, making it a relevant topic for the medical college admission test, MCAT. The hepatic portal vein plays a crucial role in supporting liver function, and understanding its relationship with blood circulation is essential for medical students preparing for the MCAT exam. The hepatic portal vein is a major blood vessel that transports blood from the digestive organs, such as the stomach and intestines, to the liver. Unlike most veins that carry oxygen-depleted blood back to the heart, the hepatic portal vein carries nutrient-rich blood that has absorbed products of digestion. Upon ingestion of food, the digestive system breaks down complex molecules into smaller nutrients. These nutrients, including glucose, amino acids, and fatty acids, are absorbed by the small intestine and then transported to the liver via the hepatic portal vein. This specialized venous system allows the liver to play a central role in regulating nutrient levels in the blood. In the liver, a series of intricate metabolic processes take place. Glucose, for instance, undergoes glycogenesis, gluconeogenesis, and glycolysis, helping to regulate blood sugar levels. Amino acids are utilized for protein synthesis and converted into various essential molecules. Fatty acids undergo beta oxidation, contributing to energy production and lipid metabolism. Furthermore, the liver detoxifies the blood by removing harmful substances and metabolizes drugs. It also synthesizes plasma proteins such as albumin and clotting factors, contributing to the maintenance of blood volume and coagulation. The intricate relationship between the hepatic portal vein and liver function highlights the organ's pivotal role in maintaining homeostasis within the circulatory and metabolic systems. This knowledge is critical for medical students preparing for the MCAT, as it forms the basis for understanding physiological processes and their implications for overall health. Structural components. Nasal cavity and pharynx. The process of respiration begins with air entering the nasal cavity, where it is filtered, humidified, and warmed. From there, air moves through the pharynx. Larynx and trachea. The larynx, commonly known as the voice box, connects the pharynx to the trachea. The trachea, in turn, branches into bronchi, leading to the lungs. Pulmonary ventilation. Inspiration and expiration. The respiratory cycle involves inspiration, inhalation, and expiration, exhalation. During inspiration, the diaphragm contracts and the ribcage expands, creating negative pressure that draws air into the lungs. Expiration occurs when the diaphragm relaxes and the ribcage contracts. Gas exchange. Alveoli. The functional units of the lungs are the alveoli, tiny air sacs where gas exchange occurs. Oxygen diffuses from the alveoli into the bloodstream, while carbon dioxide moves from the blood into the alveoli for elimination. Transport of gases, hemoglobin. Oxygen is primarily transported in the blood bound to hemoglobin within red blood cells. This oxyhemoglobin complex releases oxygen to tissues as needed. Carbon dioxide is transported in the blood in various forms, including bicarbonate ions. Regulation of respiration. Chemoreceptors. Specialized receptors in the brain and peripheral tissues monitor changes in blood gas levels. Chemoreceptors play a key role in adjusting the rate and depth of breathing to maintain appropriate oxygen and carbon dioxide levels. Respiratory control centers. Medulla oblongata and pons. The medulla and pons, located in the brainstem, house respiratory centers that regulate breathing. These centers respond to signals from chemoreceptors and other feedback mechanisms. Respiratory disorders. Asthma, chronic obstructive pulmonary disease, COPD, and pneumonia. Understanding common respiratory disorders is crucial. Asthma involves bronchoconstriction and inflammation. COPD comprises chronic bronchitis and emphysema, and pneumonia is an inflammatory lung condition. Pulmonary function tests, PFTs. Spirometry. PFTs, such as spirometry, measure lung volumes and capacities, providing diagnostic information about respiratory health. Understanding the intricacies of the respiratory system is essential for medical professionals, especially those preparing for the Medical College Admission Test, MCAT. Mastery of these concepts ensures a solid foundation in human physiology and prepares individuals for more advanced studies in the field of medicine. Question 6. Action of glucagon. Unpack how glucagon regulates blood sugar levels, contrasting its function with insulin. The forebrain is a crucial part of the human brain, responsible for various higher cognitive functions. Understanding its functions is essential for the Medical College Admission Test, MCAT. Here are some college-level information points about the functions of the forebrain in humans. Cerebral cortex functions. The cerebral cortex, the outer layer of the forebrain, is responsible for higher cognitive functions such as thinking, reasoning, and problem-solving. It is divided into 4 lobes: frontal, parietal, temporal, and occipital, each associated with specific functions. Frontal lobe functions. Motor cortex. Located in the frontal lobe, it controls voluntary movements. Prefrontal cortex. Involved in decision-making, personality, and social behavior. Parietal lobe functions. Somatosensory cortex. Processes sensory information related to touch, temperature, and pain. Spatial awareness. Responsible for understanding the spatial orientation of the body. Temporal lobe functions. Auditory cortex. Processes auditory information and is crucial for hearing. Hippocampus. Involved in the formation of new memories. Occipital lobe functions. Visual cortex. Processes visual information received from the eyes. Thalamus. Acts as a relay station for sensory information, directing it to the appropriate areas of the cerebral cortex. Hypothalamus. Regulates basic survival functions such as hunger, thirst, body temperature, and sleep. plays a role in the endocrine system by controlling the pituitary gland. Limbic system includes structures like the amygdala and hippocampus, involved in emotions, memory, and motivation. Basal ganglia plays a role in motor control and procedural learning. Dysfunction may lead to movement disorders such as Parkinson's disease. Cerebral hemispheres. The left hemisphere is typically associated with language and logical reasoning. The right hemisphere is often linked to creativity and spatial abilities. Understanding these functions will not only help with the MCAT, but also provide a comprehensive understanding of how the forebrain contributes to human cognition and behavior. Question 7. Catalyst in a chemical reaction. Introduce the concept of catalysts and their role in speeding up chemical reactions, making them essential in industry and daily life. Glucagon is a peptide hormone that plays a crucial role in maintaining glucose homeostasis within the body. Produced by the alpha cells of the pancreas, glucagon acts in opposition to insulin, another pancreatic hormone, to regulate blood sugar levels. Here is a college-level overview of the action of glucagon. Stimulus for release. Glucagon release is triggered by low blood glucose levels. When the concentration of glucose in the bloodstream drops, alpha cells in the pancreas release glucagon in response to signals indicating the need for increased glucose availability. Target tissues. Glucagon primarily targets the liver, but it also affects adipose tissue and muscle cells. The liver is a key organ in glucose metabolism and storage. Liver glycogenolysis. One of the primary actions of glucagon is to stimulate glycogenolysis in the liver. This process involves the breakdown of glycogen, a storage form of glucose, into individual glucose molecules. The liberated glucose is then released into the bloodstream, raising blood glucose levels. Gluconeogenesis. Glucagon also promotes gluconeogenesis in the liver. This process involves the synthesis of glucose from non-carbohydrate precursors, such as amino acids and glycerol. Gluconeogenesis contributes to the production of glucose even when glycogen stores are depleted. Inhibition of glycolysis. Glucagon inhibits glycolysis, the breakdown of glucose within cells. This inhibition ensures that glucose is not being utilized for energy within the liver cells, preserving it for release into the bloodstream. Release of free fatty acids. In adipose tissue, glucagon stimulates the breakdown of triglycerides into free fatty acids and glycerol. The released glycerol can serve as a substrate for gluconeogenesis in the liver, contributing to glucose production. Muscle effects. Glucagon has minimal direct effects on muscle tissue. However, its actions indirectly support glucose availability for muscle cells through the promotion of glycogenolysis and gluconeogenesis in the liver. Overall impact on blood glucose. The combined effects of glucagon result in an increase in blood glucose levels. This is essential for providing energy to cells, especially during fasting or periods of increased metabolic demand. Understanding the intricate balance between insulin and glucagon is critical for comprehending the regulation of blood glucose levels and the maintenance of metabolic homeostasis in the body. This knowledge is relevant not only for understanding basic physiology, but also for addressing clinical scenarios related to diabetes and other metabolic disorders. Question 8. Enzymes as biological catalysts. Highlight how enzymes act as catalysts in biological systems, facilitating essential biochemical reactions in the body. The liver is a vital organ in the human body with a multitude of functions. Understanding the potential harmful effects on the body, especially from a medical perspective, such as the MCAT test, involves knowledge of liver functions and associated pathologies. Here are some key points related to liver functions and potential issues. Detoxification. Normal function. The liver plays a crucial role in detoxifying harmful substances by breaking them down into less toxic forms. Potential harm. Excessive exposure to toxins, such as alcohol or certain drugs, can overwhelm the liver's detoxification capacity, leading to liver damage. Metabolism of nutrients. Normal function. The liver is involved in the metabolism of carbohydrates, fats, and proteins. Potential harm. Conditions like non-alcoholic fatty liver disease, NFLD, can result from an imbalance in nutrient metabolism, leading to fat accumulation in the liver. Synthesis of proteins. Normal function. The liver synthesizes proteins, including blood clotting factors and albumin. Potential harm. Liver diseases, such as cirrhosis, can disrupt protein synthesis, affecting blood clotting and leading to edema. Storage of vitamins and minerals. Normal function. The liver stores essential vitamins and minerals, including vitamin A, D, and iron. Potential harm. Liver diseases can impair the storage and release of these nutrients, contributing to deficiencies and related health issues. Bile production. Normal function. The liver produces bile, which aids in digestion and the absorption of fats. Potential harm. Gallstones or liver diseases can interfere with bile production, leading to digestive problems and nutrient malabsorption. Blood glucose regulation. Normal function. The liver helps regulate blood glucose levels by storing or releasing glucose as needed. Potential harm. Conditions like insulin resistance or liver diseases can disrupt this balance, contributing to diabetes or hypoglycemia. Detoxification of ammonia. Normal function. The liver converts ammonia, a byproduct of protein metabolism, into urea for excretion. Potential harm. Liver diseases can result in the accumulation of ammonia, leading to conditions like hepatic encephalopathy, affecting brain function. Understanding these liver functions and associated pathologies is crucial for medical students preparing for the MCAT, as questions related to organ function, dysfunction, and their impact on overall health are commonly tested. Question 9. Catalysts. A catalyst is a substance that speeds up a chemical reaction by providing an alternative reaction pathway with lower activation energy without itself being consumed in the process. Understanding the purpose and function of catalysts is crucial in the field of chemistry and is often tested on the MCAT, Medical College Admission Test. Lowering activation energy. One of the primary purposes of a catalyst is to lower the activation energy required for a chemical reaction to occur. Activation energy is the minimum energy input necessary to initiate a reaction. By providing an alternative, more efficient pathway with a lower activation energy, a catalyst facilitates the reaction, making it occur more rapidly. Increased reaction rates. Catalysts enhance reaction rates by effectively increasing the number of successful collisions between reactant molecules. By lowering the energy barrier, more particles possess the required energy to overcome it, leading to a higher frequency of successful collisions and an overall faster reaction rate. Specificity and selectivity. Catalysts are often highly specific to certain reactions, promoting selectivity in the products formed. This specificity is crucial in organic chemistry and biochemistry, where precise control over reaction pathways is essential for synthesizing specific compounds or biomolecules. Preservation of catalyst. Importantly, catalysts remain unchanged at the end of the reaction, allowing them to be used repeatedly. This distinguishes catalysts from other substances involved in a reaction that may be consumed or altered during the process. The ability of catalysts to participate in multiple reaction cycles makes them efficient and economically favorable in industrial applications. Environmental and economic impact. Catalysts play a significant role in green chemistry by enabling more sustainable and environmentally friendly processes. They can reduce the need for harsh reaction conditions, lower energy requirements, and minimize waste production. This aligns with the principles of sustainable chemistry and has broad implications for industrial applications. Examples in biological systems. Enzymes, which act as biological catalysts, are integral to numerous biochemical processes in living organisms. The functioning of enzymes in cellular reactions is crucial for maintaining life processes, and understanding enzyme catalysis is essential for those pursuing a career in the medical field. In summary, the purpose of a catalyst is to accelerate chemical reactions by providing an alternative reaction pathway with lower activation energy. This fundamental concept is not only pertinent to understanding chemical kinetics, but also has wide-ranging applications in various scientific and industrial domains. Question 10. Unlocking nature's secrets, catalysts in biological systems. Catalysts in biological systems play a crucial role in facilitating and regulating biochemical reactions necessary for life. Understanding these catalysts is essential for success on the MCAT, Medical College Admission Test. Here's a college-level overview of some key catalysts found in biological systems. Enzymes. Enzymes are proteins that act as biological catalysts. They enhance the rate of chemical reactions by lowering the activation energy required for the reaction to occur. Enzymes are highly specific, recognizing and binding to specific substrates. The enzyme-substrate complex undergoes conformational changes that stabilize the transition state, allowing the reaction to proceed more rapidly. Examples include DNA polymerase, which catalyzes DNA synthesis, and amylase, which breaks down starch into sugars. Coenzymes and cofactors. Coenzymes and cofactors are non-protein molecules that assist enzymes in catalyzing reactions. They often participate in the transfer of functional groups during biochemical processes. Examples include NAD+, nicotinamide adenine dinucleotide, and FA, flavin adenine dinucleotide, which are involved in redox reactions, and coenzyme A, which participates in acetyl group transfer. Ribozymes. Unlike most catalysts, ribozymes are catalytic RNA molecules. They play a role in RNA processing and catalyze specific reactions, such as the cleavage of RNA strands. Ribozymes were initially discovered in the context of RNA splicing, where they facilitate the removal of introns from pre-mRNA molecules. Metal ions. Metal ions often act as cofactors for enzymes, assisting in substrate binding or catalysis. Examples include Zn2+, Mg2+, and Fe2+, which stabilize enzyme substrate complexes or participate directly in the catalytic mechanism. Metalloenzymes, such as carbonic anhydrase, contain metal ions essential for their activity. Prosthetic groups. Prosthetic groups are non-amino acid components tightly bound to enzymes, contributing to their catalytic activity. Heme, a prosthetic group containing iron, is found in hemoglobin and myoglobin and is crucial for oxygen transport and storage in the body. Understanding the role of these catalysts in biological systems is fundamental for grasping the intricacies of biochemical processes. The MCAT often assesses this knowledge to evaluate a candidate's understanding of the molecular and cellular foundations of living organisms. Question 1. Which enzyme is responsible for the synthesis of the lagging strand during DNA replication? A. DNA ligase. B. DNA helicase. C. DNA polymerase. D. Primase. Question 2. Which white blood cell is primarily responsible for phagocytosis and plays a crucial role in the innate immune response? A. Neutrophil. B. Lymphocyte. C. Monocyte. D. Eosinophil. Question 3. Which of the following statements best describes the role of blood in the hepatic portal vein in supporting liver function? A. Hepatic portal vein carries oxygenated blood to the liver for energy production. B. Blood in the hepatic portal vein transports nutrients absorbed from the digestive tract to the liver. C. The hepatic portal vein is responsible for removing toxins from the liver and transporting them to the digestive system. D. Blood in the hepatic portal vein is primarily involved in the distribution of hormones throughout the body. Question 4. Which checkpoint in the cell cycle ensures that DNA replication is complete before entering mitosis? A. G1 checkpoint. B. S checkpoint. C. G2 checkpoint point.
Question 5. Which of the following statements regarding the respiratory system in humans is correct? A. The diaphragm contracts during inhalation, causing an increase in thoracic volume. B. Oxygen is primarily transported in the blood bound to hemoglobin within red blood cells. C. The trachea is composed of cartilage rings, providing flexibility and preventing collapse. D. Surfactant is produced by type 1 alveolar cells and helps maintain alveolar surface tension. Question 6. Which of the following statements accurately describes a function of the forebrain in humans? A. The forebrain is primarily responsible for regulating basic bodily functions such as breathing and heart rate. B. The forebrain plays a crucial role in sensory processing, including the interpretation of visual and auditory stimuli. C. The forebrain is primarily involved in coordinating motor movements and maintaining balance and posture. D. The forebrain is responsible for the production and secretion of hormones by the endocrine system. Question 7. Which of the following statements accurately describes the action of glucagon? A. Glucagon promotes glycogen synthesis in the liver. B. Glucagon inhibits gluconeogenesis. C. Glucagon enhances glucose uptake in muscle cells. D. Glucagon stimulates glycogenolysis in the liver. Question 8. Which of the following liver functions is potentially harmful to the body? A. Glycogen storage. B. Detoxification of drugs and toxins. C. Synthesis of blood clotting factors. D. Metabolism of dietary fats. Question 9. What is the primary purpose of a catalyst in a chemical reaction? A. Increase the temperature of the reaction. B. Decrease the activation energy of the reaction. C. Change the stoichiometry of the reaction. D. Slow down the reaction rate. Question 10. Which of the following statements best describes the role of enzymes as catalysts in biological systems? A. Enzymes alter the equilibrium of a chemical reaction. B. Enzymes provide energy to drive chemical reactions. C. Enzymes increase the rate of a reaction by lowering the activation energy. D. Enzymes change the reactants into completely different products. These are the answers for the test. Question 1. Which enzyme is responsible for the synthesis of the lagging strand during DNA replication? A. DNA ligase. B. DNA helicase. C. DNA polymerase III primase. Correct answer. A. DNA ligase. Explanation. During DNA replication, the lagging strand is synthesized discontinuously in small fragments called Okazaki fragments. The enzyme responsible for joining these fragments and sealing any nicks or gaps in the sugar phosphate backbone is DNA ligase. Option A is correct because DNA ligase plays a crucial role in the final step of lagging strand synthesis by catalyzing the formation of phosphodiester bonds between adjacent Okazaki fragments. It effectively ligates or connects these fragments into a continuous strand. Let's briefly discuss why the other options are incorrect. DNA helicase. DNA helicase is responsible for unwinding the DNA double helix by breaking hydrogen bonds between complementary base pairs. It is not directly involved in the synthesis of the lagging strand. C. DNA polymerase III. DNA polymerase III is primarily involved in the elongation of both the leading and lagging strands by adding nucleotides in a 5' to 3 direction. However, it is not responsible for joining Okazaki fragments or sealing nicks in the lagging strand. D. Primase. Primase is responsible for synthesizing RNA primers on the lagging strand, providing a starting point for DNA synthesis by DNA polymerase III. While primase is essential for lagging strand synthesis, it does not directly participate in joining Okazaki fragments or sealing nicks. In summary, DNA ligase is the correct answer because it performs the crucial function of connecting Okazaki fragments during the synthesis of the lagging strand. Question 2. Which white blood cell is primarily responsible for phagocytosis and plays a crucial role in the innate immune response? A. Neutrophil. B. Lymphocyte. C. Monocyte. D. Eosinophil. Correct answer. A. Neutrophil. Explanation. A. Neutrophil. Neutrophils are the most abundant white blood cells and are known for their rapid response to infections. They are phagocytic cells that engulf and digest pathogens, particularly bacteria. Neutrophils are an essential component of the innate immune system and play a critical role in the early stages of defense against infections. B. Lymphocyte. Lymphocytes are involved in the adaptive immune response and are responsible for immune memory. They include T cells, B cells, and natural killer, NK cells. While lymphocytes play a vital role in specific immune responses, they are not primarily responsible for phagocytosis. C. Monocyte. Monocytes are precursors to macrophages and dendritic cells. They are involved in phagocytosis and antigen presentation. Monocytes circulate in the blood and migrate to tissues where they differentiate into macrophages. However, neutrophils are more directly associated with phagocytosis in the bloodstream. D. Eosinophil. Eosinophils are white blood cells involved in combating parasitic infections and allergic reactions. They release toxic substances to destroy parasites, but are not the primary cells responsible for phagocytosis. In summary, neutrophils are the correct answer because they are the main phagocytic cells in the bloodstream, playing a crucial role in the immediate response to infections. Question 3. Which of the following statements best describes the role of blood in the hepatic portal vein in supporting liver function? A. The hepatic portal vein carries oxygenated blood to the liver for energy production. B. Blood in the hepatic portal vein transports nutrients absorbed from the digestive tract to the liver. C. The hepatic portal vein is responsible for removing toxins from the liver and transporting them to the digestive system. D. Blood in the hepatic portal vein is primarily involved in the distribution of hormones throughout the body. Correct answer. B. Blood in the hepatic portal vein transports nutrients absorbed from the digestive tract to the liver. Explanation. A. Incorrect. The hepatic portal vein carries deoxygenated blood from the digestive organs to the liver, not oxygenated blood. The liver's role is not primarily related to energy production from oxygenated blood. B. Correct. The hepatic portal vein collects nutrient-rich blood from the digestive organs, including the stomach and intestines, and transports these nutrients to the liver. This blood contains products of digestion such as glucose, amino acids, and other nutrients that the liver processes and regulates. C. Incorrect. While the liver is involved in detoxification, the hepatic portal vein does not transport toxins away from the liver. Instead, the liver processes and detoxifies substances that enter the bloodstream through the digestive system. D. Incorrect. The hepatic portal vein is not primarily responsible for the distribution of hormones. Hormones are generally transported through the systemic circulation, not the hepatic portal vein. In summary, option B is the correct answer because the hepatic portal vein plays a crucial role in transporting nutrients from the digestive system to the liver for processing and regulation. Which checkpoint in the cell cycle ensures that DNA replication is complete before entering mitosis? A. G1 checkpoint, B. S checkpoint, C. 2 checkpoint, D. Correct answer, C. G2 checkpoint. Explanation: the G2 checkpoint, also known as the DNA damage checkpoint, occurs at the end of the G2 phase of the cell cycle, just before the cell enters mitosis. The primary purpose of this checkpoint is to ensure that DNA replication is complete and that the DNA is undamaged before the cell proceeds to divide. The cell checks for any DNA damage or incomplete replication, and if issues are detected, it can halt the cell cycle to allow for repair. Reasons for incorrect options. A. G1 checkpoint. The G1 checkpoint primarily checks for cell size, nutrient availability, and DNA damage. It does not specifically assess whether DNA replication is complete. B. S checkpoint. The S checkpoint, occurring during the synthesis phase, monitors DNA replication. However, it doesn't ensure that replication is completed accurately. It mainly assesses if the DNA synthesis is initiated. D. M checkpoint. The M checkpoint, also known as the spindle checkpoint, occurs during mitosis and ensures proper chromosome alignment and attachment to the spindle fibers. It doesn't specifically check for DNA replication completeness. In summary, the G2 checkpoint is crucial for ensuring that the cell has accurately completed DNA replication before entering mitosis, making option C the correct answer.
Question 5. Which of the following statements regarding the respiratory system in humans is correct? A. The diaphragm contracts during inhalation, causing an increase in thoracic volume. B. Oxygen is primarily transported in the blood bound to hemoglobin within red blood cells. C. The trachea is composed of cartilage rings, providing flexibility and preventing collapse. D. Surfactant is produced by type 1 alveolar cells and helps maintain alveolar surface tension. Correct answer: B. Oxygen is primarily transported in the blood bound to hemoglobin within red blood cells. Explanation: incorrect. The diaphragm contracts during inhalation, but this action results in a decrease in thoracic volume, creating a pressure gradient that allows air to enter the lungs. B. Correct. Oxygen is transported in the blood primarily by binding to hemoglobin within red blood cells. Hemoglobin forms a reversible bond with oxygen, allowing for efficient transport from the lungs to tissues. C. Incorrect. The trachea is indeed composed of cartilage rings, but these rings provide structural support and prevent collapse, not flexibility. D. Incorrect. Surfactant is produced by type 2 alveolar cells, not type 1. Additionally, surfactant reduces surface tension in the alveoli, preventing their collapse, not maintaining surface tension. In summary, the correct answer is B because it accurately describes the primary mode of oxygen transport in the bloodstream, while the other options contain inaccuracies about respiratory system physiology. Question 6. Which of the following statements accurately describes a function of the forebrain in humans? A. The forebrain is primarily responsible for regulating basic bodily functions such as breathing and heart rate. B. The forebrain plays a crucial role in sensory processing, including the interpretation of visual and auditory stimuli. C. The forebrain is primarily involved in coordinating motor movements and maintaining balance and posture. D. The forebrain is responsible for the production and secretion of hormones by the endocrine system. Correct answer. B. The forebrain plays a crucial role in sensory processing, including the interpretation of visual and auditory stimuli. Explanation. A. This statement is incorrect. The regulation of basic bodily functions such as breathing and heart rate is primarily associated with the brainstem, not the forebrain. B. This is the correct answer. The forebrain, specifically the cerebral cortex, is responsible for sensory processing and the interpretation of various stimuli. Visual and auditory information is processed in specialized regions of the cerebral cortex. C. This statement is incorrect. Coordinating motor movements and maintaining balance and posture are functions associated with the cerebellum, which is located in the hindbrain, not the forebrain. D. This statement is incorrect. The production and secretion of hormones by the endocrine system are primarily regulated by structures such as the hypothalamus and pituitary gland, which are located in the diencephalon, part of the forebrain. But the statement oversimplifies the functions of the forebrain. In summary, option B is the correct answer because the forebrain, specifically the cerebral cortex, is involved in sensory processing and the interpretation of visual and auditory stimuli. Question 7. Which of the following statements accurately describes the action of glucagon? A. Glucagon promotes glycogen synthesis in the liver. B. Glucagon inhibits gluconeogenesis. C. Glucagon enhances glucose uptake in muscle cells. D. Glucagon stimulates glycogenolysis in the liver. Correct answer. D. Glucagon stimulates glycogenolysis in the liver. Explanation. A. Incorrect. Glucagon does not promote glycogen synthesis. It promotes the breakdown of glycogen, glycogenolysis, to release glucose into the bloodstream. B. Incorrect. Glucagon actually stimulates gluconeogenesis, the process of producing glucose from non-carbohydrate sources such as amino acids and glycerol. C. Incorrect. Glucagon does not enhance glucose uptake in muscle cells. Its primary role is to raise blood glucose levels by promoting the release of glucose from the liver. D. Correct. Glucagon stimulates glycogenolysis in the liver, breaking down stored glycogen into glucose. This process helps elevate blood glucose levels, especially during periods of fasting or low blood glucose. In summary, glucagon's main actions include promoting glycogenolysis in the liver and stimulating gluconeogenesis, both of which contribute to increasing blood glucose levels. Question 8. Which of the following liver functions is potentially harmful to the body? A. Glycogen storage. B. Detoxification of drugs and toxins. C. Synthesis of blood clotting factors. D. Metabolism of dietary fats. Correct answer. B. Detoxification of drugs and toxins. Explanation. The liver plays a crucial role in detoxifying various drugs and toxins in the body. This process involves enzymes in the liver metabolizing these substances to make them more water-soluble, allowing for their excretion from the body. While this detoxification function is essential, it can also be potentially harmful if the liver is exposed to high levels of toxic substances, leading to liver damage or dysfunction. Incorrect options. A. Glycogen storage. The liver's glycogen storage function is not harmful. It is a vital process that involves storing glucose for energy production during times of need. C. Synthesis of blood clotting factors. The synthesis of blood clotting factors by the liver is essential for proper blood coagulation and is not inherently harmful. D. Metabolism of dietary fats. The liver's role in the metabolism of dietary fats is crucial for energy production and nutrient storage. While excessive fat metabolism can lead to health issues, it is not the primary harmful function of the liver. In summary, option B is correct because the detoxification function of the liver, while necessary, can become harmful under conditions of excessive exposure to drugs and toxins. Question 9. Which of the following liver functions is potentially harmful to the body? A. Glycogen storage. B. Detoxification of drugs and toxins. Synthesis of blood clotting factors. Metabolism of dietary fats. Correct answer. Detoxification of drugs and toxins. Explanation. The liver plays a crucial role in detoxifying various drugs and toxins in the body. This process involves enzymes in the liver metabolizing these substances to make them more water-soluble, allowing for their excretion from the body. While this detoxification function is essential, it can also be potentially harmful if the liver is exposed to high levels of toxic substances, leading to liver damage or dysfunction. Incorrect options. A. Glycogen storage. The liver's glycogen storage function is not harmful. It is a vital process that involves storing glucose for energy production during times of need. C. Synthesis of blood clotting factors. The synthesis of blood clotting factors by the liver is essential for proper blood coagulation and is not inherently harmful. D. Metabolism of dietary fats. The liver's role in the metabolism of dietary fats is crucial for energy production and nutrient storage. While excessive fat metabolism can lead to health issues, it is not the primary harmful function of the liver. In summary, option B is correct because the detoxification function of the liver, while necessary, can become harmful under conditions of excessive exposure to drugs and toxins. Question 10. What is the primary purpose of a catalyst in a chemical reaction? A. Increase the temperature of the reaction. B. Decrease the activation energy of the reaction. C. Change the stoichiometry of the reaction. D. Slow down the reaction rate. Correct answer, B. Decrease the activation energy of the reaction. Explanation. A catalyst is a substance that speeds up the rate of a chemical reaction without being consumed in the process. The correct answer is B because a catalyst works by lowering the activation energy required for the reaction to proceed. By reducing the activation energy, a catalyst provides an alternative reaction pathway with a lower energy barrier, allowing more reactant molecules to overcome the barrier and form products. This acceleration of the reaction does not alter the thermodynamics or overall energy change of the reaction. Let's examine why the other options are incorrect. A. Increase the temperature of the reaction. Increasing the temperature can enhance reaction rates, but it is not the primary purpose of a catalyst. A catalyst can facilitate reactions at lower temperatures. C. Change the stoichiometry of the reaction. A catalyst does not alter the stoichiometry of the reaction. It affects the reaction kinetics, but not the overall chemical equation. D. Slow down the reaction rate. This is the opposite of the purpose of a catalyst. A catalyst is intended to increase the reaction rate, not slow it down.
Question 10. Which of the following statements best describes the role of enzymes as catalysts in biological systems? A. Enzymes alter the equilibrium of a chemical reaction. B. Enzymes provide energy to drive chemical reactions. C. Enzymes increase the rate of a reaction by lowering the activation energy. D. Enzymes change the reactants into completely different products. Answer: C. Enzymes increase the rate of a reaction by lowering the activation energy. Explanation: enzymes are biological catalysts that facilitate chemical reactions within living organisms. The correct answer is C, because enzymes lower the activation energy required for a chemical reaction to occur. Activation energy is the energy barrier that must be overcome for a reaction to proceed. By lowering this barrier, enzymes accelerate the rate of reactions without being consumed in the process. Let's examine why the other options are incorrect. A. Enzymes altering the equilibrium of a reaction is not a typical role. Enzymes do not change the position of equilibrium, they only accelerate the attainment of equilibrium. B. Enzymes do not provide energy for reactions. They facilitate reactions by lowering the activation energy, but they do not generate energy. D. Enzymes do not change the reactants into completely different products. They facilitate reactions by providing an alternative pathway with lower activation energy, but they do not alter the fundamental chemical nature of the reactants. In summary, the correct answer is C, because enzymes act as catalysts by lowering the activation energy and increasing the rate of biological reactions. Yo, it's the med student life, but it's feeling strange, caught up in this world where emotions rearrange. Stethoscope round my neck, but my heart feels numb. In the ER, but I'm feeling so glum. Patients come and go, but I'm detached inside, trying to save lives, but my feelings, they hide. Cadavers on the table, but they don't make me flinch. In this clinical world where emotions inch. But behind this facade, there's a storm brewing deep. Trying to suppress, but it's hard to keep. A wall around my heart, like a fortress strong, but inside I'm yearning to find where I belong. In the hustle and the grind, but I'm feeling detached. Emotions on lockdown, like they've been mismatched. In this medical journey, where I'm searching for my track, I'm just a med student trying to find my way back. Rounds in the ward, but I'm feeling so cold. Like a robot on autopilot, my emotions on hold. Sympathy and empathy, they seem so far away, in this clinical maze where emotions decay. Diagnosing diseases, but what about the soul? In this sterile environment where feelings take a toll. But I'll push through the detachment. Find my humanity in this medical odyssey, searching for clarity. Behind the mask of professionalism, there's a beating heart, yearning to connect, to play its part. In this sea of white coats, I'll find my emotion and break free from this detached commotion. In the hustle and the grind, but I'm feeling detached, emotions on lockdown like they've been mismatched. In this medical journey where I'm searching for my track, I'm just a med student, trying to find my way back. So I'll peel back the layers, let the feelings flow, in this medical world where empathy must grow. Though detachment may linger, I'll break free and embrace the emotions that make me me. Yo, it's the med student life, but I'll find my ground in this journey of healing where emotions abound. No longer detached, but connected and true. In the world of medicine, I'll find my breakthrough. Meaningful scriptures from the Word of God to think about. John 4:24, NIV, God is spirit, and his worshippers must worship in the spirit and in truth. Ephesians 3:20-21, MSG, God can do anything, far more than you could ever imagine or guess or request in your wildest dreams. He does it not by pushing us around, but by working within us, his spirit deeply and gently within us. Glory to God in the church. Glory to God in the Messiah, in Jesus. Glory down all the generations. Glory through all millennia. Oh, yes, a few insights from the Word of God to meditate on. John 5:16-20, NLT. 16. So the Jewish leaders began harassing Jesus for breaking the Sabbath rules. 17. But Jesus replied, "My Father is always working, and so am I." So the Jewish leaders tried all the harder to find a way to kill him. For he not only broke the Sabbath, he called God his Father, thereby making himself equal with God. 9. So Jesus explained, "I tell you the truth, the Son can do nothing by himself. He does only what he sees the Father doing. Whatever the Father does, the Son also does." 20. For the Father loves the Son and shows him everything he is doing. In fact, the Father will show him how to do even greater works than healing this man. Then you will truly be astonished. Survival question alert. What foods fight inflammation in your body, and how do you prepare them? Which biological concept did you find most intriguing from this video? Please comment down below. How would you explain the importance of DNA replication to someone unfamiliar with the topic? Please comment down below. Are there specific organ functions or physiological processes you would like us to explore in future episodes? Please comment down below. Thank you for joining us on this journey through the wonders of biology. Be sure to subscribe for more insightful content. Hello there, I am Angelique Granville, and I'm a professional listener, digital products entrepreneur, and content creator. I have a degree in biology from Adelphi University, and I am passionate about helping allied health professionals succeed. I am here to help you get into medical school. On this channel, I share my tips and hacks on how you can pass your entrance exams so you can get into medical school. Do you want to talk to me for an hour about your life and your problems? Check out my therapy programs. The link is in the description. It says, "Let me help you in life." If you are getting value from this content, I urge you to like, subscribe, and share this video with all of the pre-medical students, biology students and majors, physics majors and engineering majors that you know. If you know anyone who is struggling to study for the MCAT, please tell them about the Hospital Near Me Medical Career Training channel. Together, we can conquer you getting into medical school and feel better about being accepted by the college of my choice. Please hit the notification bell so that you never ever miss another one of my videos. If you haven't already liked this video, make sure to give it a big thumbs up and send this video to at least 3 people. I know you can think of them. If somebody pops in your head right now and send it to them, WhatsApp, Facebook, TikTok, LinkedIn, and Instagram. Challenge them to do it with you, and you can get results together. Come and take my easy MCAT exam. If you are getting value from this channel, all I ask is that you let my ads play on the video. And if you could bless my PayPal, Monzo, Cash App, Venmo, Buy Me a Coffee, GoFundMe, Patreon, or give me a thank you donation of $1 to $5. or with a donation of any amount, it would be greatly appreciated. I am saving up for a new home as my old one got severely damaged in Hurricane Sandy and left me homeless. I want to thank you for watching the Hospital Near Me Medical Career Channel for allied health professionals. I look forward to being a part of your medical school journey and being a part of your own growth. Walk with God. Thanks a bunch for watching this video. Please like, comment, and subscribe to Hospital Near Me Medical Career Training. We are your source for medical college training. Thanks again, and stay tuned for the Shopping Network. Here is some useful information for you, and I quote, Long Island Coalition for the Homeless, their phone number is 631-464-4314, extension 1118. The helpline is a connection to resources and support for people experiencing homelessness. The helpline is a messaging system for people experiencing homelessness to connect with a community support specialist. The helpline is also available through electronic form at www.liehomeless.org and under the Need Help tab. An access point for coordinated entry for Nassau and Suffolk counties. The helpline is not a 24/7 hotline. It is not a crisis response line. It is not direct access to shelter, as emergency shelter can only be accessed through Department of Social Services. It is not a referral source for permanent housing programs. The helpline offers peer support, help with problem solving, resource navigation, help with development of a housing plan to exit homelessness, connection to the street outreach team or other community resources for people living unsheltered, on the street or place not meant for human habitation. Domestic violence support connections provided by the Safe Center of Long Island, 516-542-0404. On Instagram, they are @liehomeless and DSS or Welfare After Hours lines in Nassau, 516-573-8626, and Suffolk, 631-854-9100.